Rustin Lowe. Rustin Lowe is graduating with a degree in biology and administration in medical sciences from Brandon, Florida, been at SMU for 3.5 years. During this time, he's made the Dean's List. He's TA for me in organic chemistry for a year and a half. He served as Vice President of HSO. He's a member of who he played for. He's done chemistry research under my supervision for 2 years and went to many conferences, national and local, and took part in the research residency program at Lifton Regional Health, which you probably heard his presentation this morning. After graduating, he will be attending Lincoln Memorial University College of Osteopathic Medicine, where he's anticipated to graduate in 2028, pursue interest in radiology and anesthesiology. Without any further ado, let's welcome Rustin Lowe to speak about his research. Thank you very much.
I need the mic. Awesome. Well, thank you very much. As you said, I'm Weston Logue, and today I'm going to be talking on the synthesis of a dithiocarbamate peptidomimetic scaffold as a potential anti-cancer agent. So just to get started, let's look at some just cancer and its development as of today. Cancer continues to lead the world in cause of death, only behind cardiovascular disease, and has claimed the life of 609,000 people in 2022, just in the United States alone. That does not include worldwide. And it's extremely complex and variable among many different organ systems, and remains one of the most funded areas of research annually for the past several years. And in 2023, the Consolidated Appropriations Act allocated $7.3 billion to the National Cancer Institute for the fiscal year of 2024. So it is continually being increased in funding. Let's talk about a little bit of cancer's complexity. But as I said before, it varies from organ system to organ system, and there are many different kinds of factors that contribute to cancer growth and its metastasis. One of such is epigenetic mutations, oncogene activations, apoptosis evasion, somatic mutations, tumor suppressor deactivations, and there are much more different factors that contribute to the ongoing of cancer. And so I'm going to talk through this figure here just for a moment. Kind of similar to those mutations here is what begins the metastasis and cancerous development of these normal tissues, which then would progress into these cancer progenitor cells, which then these growth of transformed cells can then be also altered through many different transcription factors, other epigenetic alterations and mutations, which then lead to epithelial mesenchymal translation, transition, which is a fancy way of saying these cancerous cells are becoming mobile. They're starting to breach through the basement membrane of that organ and transport into the bloodstream to other organs, which is happening here of these growth of metastatic cancer cells. Current therapies, there are many different therapies and no sure treatment of every kind of cancer and cell line out there. But as of therapies that we have right now, include surgery, chemotherapies, stem cell therapies, immunotherapies, ablation therapies, gene therapies, and many others. And so with that, there's still a need for much research in cancer, and that is where I propose a synthesis and characterization of a dithiocarbamate scaffold structure, and to evaluate the effectiveness of the peptidomimetic on biological assays in effort to create a novel anti-cancer agent therapy. And just for starters, this is what we're looking at for this dithiocarbamate molecule. And I'll get into what exactly a peptidomimetic is in just a moment, but the important structure that we're looking for is right there with those two sulfurs and a carbon double bond sulfur to the amine group, which is this group right here. So getting into what exactly a peptidomimetic is, it's a very fancy way of saying it's an artificial peptide bond, which basically means we have a normal peptide bond here, and then we can do different alterations to these backbones, which make it resistant to many different kinds of proteolysis, makes it much more orally available, and easy to synthesize. With that, my methods for this peptidomimetic include reduction of amino acids by sodium borohydride and iodine, which is what this figure here is showing, where you have a carboxylic acid being reduced under these conditions to an alcohol. And then we benzylate the nitrogen group and bromination using thionyl bromide, which is this figure here showing this benzylated nitrogen, which serves as our protecting group, and then undergoing bromination of that alcohol. And preliminary diethyl carbamate synthesis is what we carried out next to kind of test and make sure the synthesis was going to go over well. And then coupling of the prepared isoleucine and isoleucine methyl ester salt. And that prepared isoleucine is the reduced and brominated and nitrogen isoleucine that we prepared. And this went through coupling again through a dipeptide coupling with the prepared glycine and which is the tripeptide, and then biological assay screenings with negative DMSO and positive controls, which is Venticlax, which I will get into in a moment. So what's the target that we're looking at with this peptidomimetic? It's a specific pathway called the BCL2 back pathway, and it's a specific protein-protein interaction. And in this figure, we show a inactivated BH3 complex, which will then can be activated by these anti-apoptotic and anti-autophagic proteins and factors, which then could inhibit the apoptosis. But then they would need to be activated BH3, which could then activate back, metabolizing into the mitochondria, then leading through apoptosis of the cell. And getting into that a little bit deeper, the pathway for this starts with a cytotoxic stimuli of a growth factor being inhibited or a cancer cell being tagged for apoptosis and death, would proceed through BH3 group, then activating back, and then once back is metabolized, it's mitochondrial release of cytochrome C, going through APAF1, releasing caspase 9, and there's the caspase 9 cascade leading to apoptosis of the cell. And so the back peptidomimetic computation that was analyzed over at Moffitt Cancer Center, this is that image here with the complete amino acid sequence, which adopts into this, not necessarily alpha helical structure, but this helical structure in nature, which is thought to be this crucial binding region of back protein complex, which we're hypothesizing can activate that important BH3 complex, leading to apoptosis of the cancerous tissue. And these very important interactions between these amino acids that we have placed here are hypothesized or to lead to the activation of BAC leading to apoptosis. This is a graph showing the BCL2 expression in the specific cancer of acute myeloid leukemia, which as you can see here in the newly diagnosed, we see a high increase in upregulation of BCL2 expression compared to the control of normal healthy patients. This carbamatoid scaffold was first synthesized in the lab at Dr. Salvatore. And just to give a general overview of how this was synthesized, an isoleucine methyl ester salt form underwent cesium carbamate and carbon dioxide synthesis, which then promoted the coupling for the first dipeptide, where you have the protecting group of benzene, that bromination that I was talking about earlier, which then would be taken off under hydrogenation under pressure. And then eventually the exact same synthesis would proceed through, this time with a leucine compound, and would afford the first tripeptide. So I'm going to talk about this current BCL2 inhibitor, which is relatively new in the past several years. It's called Venticlax. And Venticlax is in 2016 became the very first FDA-approved BCL2 inhibitor for the specific treatment of chronic lymphocytic leukemia. And during trials, this was also known as ABT-199. And this BCL2 inhibitor acts as a direct BH3 mimic, which is different than what we are proposing as a direct back mimic of that crucial sequence I was talking about earlier. And these studies have shown that there is a buildup resistance and prolonged higher dosage over long periods of time with this Venticlax. And this is the structure of Venticlax and the current sample that we have in our lab to be tested as our positive control, which I'll get into in the assay. So with that, our current focus leads to my specific aim of the synthesis and characterization of a diethyl carbamate peptidomimetic molecule. And so you may be asking, why even look at diethyl carbamates to begin with? Why? It's a strange molecule. Why would we be interested in this at all? And the reason is there's a lot of papers showing that there's really good treatment of bacterial and fungal infections proteasome inhibition. It's currently in phase 2 clinical trials for a potential AIDS treatment and obviously potential cancer therapy with another phase 2 clinical trial of a diethylcarbamate molecule for the treatment of hepatocellular carcinoma or liver cancer. So this is my proposed diethylcarbamate scheme, which as you can see is very similar to the carbamatoid scheme seen on the previous slide, where we have the same isoleucine methyl ester salt form proceeding through a cesium carbamate reaction, where instead of the carbon dioxide reagent, we have a carbon disulfide reagent, which puts those two sulfur groups with that double bond carbon and here in those R groups. And it proceeds through the same way with the brominated and benzylated isoleucine, and then which would be the protecting group would be taken off with hydrogenation. However, we have been told at previous conferences that we're not sure how this sulfur and thiol groups are going to react with this palladium catalyst there, which could potentially poison the reaction, where we would need to kind of go back and maybe think about changing this protecting group to something else. So the synthesis and characterization. First, we prepared the sequence amino acids with the protecting group and the bromination. Then we carried out a preliminary synthesis of a diethylcarbamate structure just to make sure we knew we could go through this synthesis process. And this is that first preliminary screening of a diethylcarbamate molecule, which we sent through infrared spectrometry. And this is that area that we could confirm that we had that carbon double bond to the sulfur group there. And then after we knew that we had that, we moved on to the synthesis of our prepared isoleucine and isoleucine salt form, which then was this product here, kind of this very thick brown yellowish oil, which we compared the infrared spectrometry of this one, and we confirmed that it does have the exact same peaks around 1300 and 1400 with this one. And that leads to my specific aim 2, where we subject the de novo peptide-like diethylcarbamate to biological assays for screening of biological activity, and then eventually tumor selectivity. So in our biological screening, we would prepare to use agar gels using the Kirby-Bauer disc diffusion method, testing for bacterial inhibition of E. coli. And then we would have that compared to our positive control, which would be Venticlax, which is the FDA-approved drug I talked about earlier, and a negative control, which would be used in DMSO. And then eventually leading to cell culture screening, which we can use with KOPT-K1 and K562, which KOPT-K1 is a lymphocytic leukemia, and K562 is a popular cell line for acute myeloid leukemia. And these are my references. And I would just like to thank Dr. Ralph Salvatore for being the lead on this project and all the help that he's been to me over my time here. The peptidomimetic research team, Southeastern University and our science department, University of South Florida's chemistry department, and the H. Lee Moffitt Cancer Center and their drug discovery department for all they've done to help. And I can now take any questions. Yes.
So DPCs are already available commercially as fungicides and for other purposes, correct?
What is it? Sorry.
DPC, your diethylcarbamate.
Oh, yes, yes.
DPCs are already available.
Yes, they are.
Fungicides.
Yes.
So what's the point, or how is it different in your synthesis? Like, what are you trying to accomplish? Is it totally different chemically?
Yeah, it's a very different structure. The main diethylcarbamate it was at the beginning in my primary objective with that same structural functional group there, which is carried out in many different drug molecules. We just hypothesized that if we put that same functional group in our scheme of our amino acid sequence, that it would have enhanced anti-tumor and apoptosis effects on it.
Okay, so you're changing based on what you're going to actually attach.
Yes, yes.
You have not done into that part yet.
We have two coupled right now with that, and that was the IR spectrums that I showed was with the dipeptide molecule. We haven't gotten to biological testing yet. We did have. Yes, we do know it has the structure.
Okay, now those are good. It's really, really cool. So my next question to that is, when you do that for application, for example, so I love the idea you're doing a de novo with it, but why bacteria? And bacteria doesn't exercise cancer.
Yeah.
cancer, why not heal them? Why not actually cancer cells because they're available?
Right. Yeah, we eventually do want to move towards that. We thought the idea behind the first bacterial testing was just to compare it to venetoclax biological activity. And we had this done in biochemistry 2 lab where we took a dipeptide and we used the Kirby-Bauer method and tested it against that. And that's kind of what sparked the idea of like, why don't we do this with our dipeptide? And so that was the idea behind that, just to see some sort of bacterial. Right, it's not cancer.
It's been shown that
Right.
it's antibacterial, antifungicidal, right? Like, I mean, fungicide, so.
Right.
Last one. So if you are to apply this in humans for cancer cells, it's been shown that it will actually metabolically become an ethylene thiourea, the ETU, which is toxic, neurotoxic in the process. So have you read along those lines, like what would be, like, and that's the reason I asked at the beginning. I don't know how it's going to be metabolically transformed since you're transforming the molecule compared to the actual UTC,
Right.
dithylcarbamate. That might be one very important thing you need to look into because the neurotoxicity of the byproduct of the original UTC is established.
Okay. Right.
And so if you can show that you're changing that, you might not be able to produce that ECU.
Right.
Based on the,
Whatever changes it
so
went
that
through.
would be a very good.
Yeah, thank you. That is a good point. Anybody else?
What do you propose as an alpha helical structure? How did you?
So that was done at Moffitt with the computational analysis, and that's how they came out with this alpha helical or helical structure of what they proposed as how this original peptidomimetic would conform to.
Do you think the sulfur analog?
Potentially, depending, because those sulfur analogs are much bigger, especially since we have 2 thiol groups, that we don't know exactly how this alpha helical structure is going to, either it could enhance the helical structure of it, or it could deter it based off what kind of bonding is interacting with the sulfurs there.
Is it linked as a disulfide bridge, or is it?
It could be.
Sulfur that are just
Right.
different. How is it working?
In the molecule itself, they're not a disulfide bridge. However, that's not saying, though, in vivo, when it's in there, that it could conform to some sort of disulfide between the 2 backbones of it.
Or maybe with an enzyme.
Yeah, maybe with an enzyme.
Do the disulfide bridge.
Right.
So we're actually going to do the cell lines, but we're not sure exactly the IRB approval. But we don't think so.
Well, yeah, I'm not sure where we are on that.
The next
An
question is.
institution. But you can do that, you're doing that at Moffitt. You should be able to do
Yeah,
that.
They could do that
at
there.
Right,
Yeah.
we were hoping here. My last question to you is you characterize the dithylcarbamate by IR, perfectly fine. Can you do it by looking at C13 and?
Yeah, we did run C13 and proton.
Where is the C double bond?
That one was pretty far downfield, around 200, whereas especially in the C13, and we did have that peak on there. I didn't include it just because I was running out of space, but we did have that on there. It was just a very small peak, so we could confirm that it was on there. If we were able to use a higher field NMR at Moffitt or at USF, it could potentially have a much stronger catch on that carbon double bond sulfur. Yes. Within Florida cancer deaths? I'm sure there is. I don't think I have specific Florida numbers, but in the US alone, there was over, what did I say at the beginning? I think about 600,000 deaths in the United States alone just last year.
Last question is, why leukemia? How did you know leukemia?
Yeah, so the original structure of that carbamatoid scaffold went through biological testing, and Moffitt Cancer came back and said this was biologically active against acute myeloid leukemia. So that's initially why we sparked an interest in pursuing that specific cell line of acute myeloid leukemia and hoping that it could be even more biologically active against it. Yes. If it's hereditary? Yes, I believe these cancer cell lines can be hereditary a lot of times. In a previous image, I had the factors that can contribute to this specific cancer metastasis, and one of those is hereditary and different genetic mutations that can happen there.
Thank you so much for running a little late.
Awesome, thank you. No, sorry. I am actually going to head out. Oh my gosh, I keep doing this is for.
Yeah, you can sit there.
Okay, our next speaker is Brooke Henry, graduated in marine biology with concentration in chemistry. She's from Florida, Florida, has been at SU for 3.5 years. While at SU, she was part of the SU indoor and beach volleyball teams as faith on fire, so worship leader and organizational leader. On graduation, she plans to attend dental hygiene school, become a dental hygienist, and possibly continue her education at dental school. Without further ado, let's welcome Brooke.
Good morning. How many of you have had a fluoride treatment at the dentist office? Okay, a lot of us. How many of you have had a cavity? A lot of us. So even with fluoride treatments, approximately 2.3 billion people still experience dental caries in their adult teeth. And then on top of that, also with fluoride treatments, an overexposure, we still see 23% of total population experiencing dental fluorosis from ages 6 to 49. And then most importantly, they're seeing a negative 0.45 mean difference in IQ score due to excessive fluoride exposure, which can be equal to approximately 7 IQ points. So then in order to understand this, we must first understand enamel. So enamel is the most mineralized and most resistant tissue, and it protects the dentin and the pulp chamber as seen in this image. And it's made up of hydroxyapatite crystals, which are made of phosphorus, calcium, and hydroxyl ions. And these hydroxyl ions have the ability to be replaced by either fluoride, carbonate, or chloride ions. So for enamel formation, the process is called amylogenesis, if it will click. And it is done with, oh my goodness, amyloblasts, which are the secretory cells that create enamel. And it has two stages, both secretory and maturation stage. In the secretory stage, the amyloblasts secrete the different proteins, amylogenin, amyloblastin, and in the second stage, the maturation stage, the hydroxyapatite crystals grow and they thicken, which then creates the enamel structure. But the amyloblasts are acellular, so they cannot regenerate, which means it's really easy for demineralization to occur. So what is demineralization? Demineralization is when calcium and phosphorus leave the tooth, as you see in this image, causing dental caries or cavities to form. And it basically is when the bacteria in the mouth breaks down the hydroxyapatite crystals and eats away at the enamel. And the opposite of this would be remineralization, which when calcium and phosphorus go back into the tooth, this can allow the enamel to strengthen and remineralize the tooth, avoiding caries and erosion. And two of the chemicals that can assist in remineralization is xylitol and fluoride. Fluoride, which we know is commonly used, and xylitol is a 5-carbon sugar polyol that can be used. So to compare fluoride and xylitol, xylitol is an electronegative anion. Both fluoride is an electronegative anion, and both xylitol and fluoride are highly soluble. Xylitol is a 5-carbon sugar polyol, as you can see by its chemical structure, C5H12O5. And for their processes and uses, fluoride can be used like I said, very commonly, and it replaces the hydroxide ions in hydroxyapatite, forming fluorapatite, and it can enhance the growth rate and provide enamel stability. Whereas xylitol can bind with the calcium ion and disrupts the process of the bacteria in the mouth, preventing it from breaking down the tooth and building plaque. And it also increases salivary flow, which helps in the remineralization process. So some cons about fluoride and xylitol. As I said earlier, fluoride has a lot of health issues, one of them being that it causes dental fluorosis, and then more importantly, a decrease in children's IQ because of overexposure. With xylitol, the issues are that it's in low concentration, so there's very minimal amount available, and the processes that we use today are very lengthy and very expensive. With that being said, xylitol is present in fruits and vegetables and many other produces, so it can be extracted from these fruits and vegetables. As you can see, there's a bunch of different produces that have xylitol, one of the highest being strawberries. This is important because in Florida, strawberry is a commodity, so it's very easily accessible and very popular. And spinach and cauliflower are also grown in Florida. But the problem with this is that fruits and vegetables are easily perished. Because of that, it's important to note that they're easily perished because we can then use it to back up my specific aim one, where I use perished fruits and vegetables in order to figure out or determine the quantitative analysis of how much xylitol is present in specifically strawberries, raspberries, cauliflower, and spinach. And then my null hypothesis for this would be that there is no difference between the fruits and vegetables of the amount of xylitol extract. So in order to do this, my method is going to start with washing the fruit. Okay, it's going to start with washing the fruit, drying it, and weighing out 3 samples of each fruit and vegetable. And then I'll blend them up and add ethyl acetate from a ratio of 1 to 10 to it and let it sit in a flask for approximately 3 hours. And then after I do that, I'm going to filter it out to separate the xylitol using a Buchner funnel and a filtration system. And once I do that, I'll put it through the rotovap to separate the xylitol from the mixture, and then use the NMR to make sure that it has the right properties of xylitol in it. And then after that, I will compare the amount of xylitol extracted from each fruit and vegetable sample and just document which one had the most xylitol present. So for the expected results, I do expect the strawberry to have the most amount of xylitol present, as it showed in the previous graph, that it had the highest amount. And cauliflower would have the most for the vegetables. Oh, one thing I want to show. I do also expect that the perished fruit and vegetables will have a higher amount of xylitol extract than the fresh fruit and vegetables. And this is because the fruits and vegetables, when they're ripening over time, the breakdown of starch into simple sugars increases the amount of xylitol present in the fruits and vegetables, as well as the enzymatic hydrolysis of xylose by yeast and bacteria for the vegetables will cause a higher amount in the vegetables. So that's why it's expected that the perished fruits and vegetables will have more than the fresh produce, which is beneficial because nobody really likes to eat not rotten, but perished fruits and vegetables. Like personally, I don't like soggy raspberries or soggy strawberries. So this will make use of the fruits and vegetables that we don't want to eat and allow it to go to something good. And then just for quick discussion, like I said, it's xylitol is occurring in fruits and vegetables and higher in strawberries and cauliflower. And because of this, a null hypothesis would be rejected. So once I get the xylitol extracted from the fruits and vegetables, the highest being strawberries, I'll take that xylitol extract and use it to do my specific aim 2, which is to test the efficacy of naturally extracted xylitol from the perished produce on enamel remineralization. And the null hypothesis for this would be that there is no difference in the testing of the purely extracted xylitol in comparison to toothpaste with fluoride in it, toothpaste with xylitol in it, and control groups. So for my method for this, I'm going to collect multiple typodonts, specifically 15 of them, because I'm going to have 3 for each group, and I have 5 different groups. So I'll have 15 of them, and I specifically chose the central incisor right because this is the first tooth that comes in contact with food and it's the most cosmetically seen. So I figured it was the most important for us if we were to test it because we want pretty teeth to be seen. And then I would need electronic toothbrushes, the Xylowhite toothpaste, Sensodyne toothpaste, which has the fluoride in it, and 3 bottles of artificial saliva to reenact the mouth process with saliva in it. So in my different groups, I'll have the 2 control groups. As I said, one will be brushed with distilled water, and one of them will have no brushing at all to represent a positive control group and a negative control group. And for my experimental groups, I'll have one that's tested with the xylitol toothpaste, one with the fluoride toothpaste, and then my xylitol extract. So for the testing of this will be done using the xylitol extract. I'm going to apply it with a Q-tip directly to the typodonts, and I'm going to let it sit on the typodonts for 2 minutes before I rinse it and brush it with distilled water. And after that, I'll place it in a petri dish with the artificial saliva and have it sealed and just let it sit until the next treatment. So this will be done twice a day, both in the morning and at night, and it will be done for approximately 12 weeks. And the same process is said for the xylitol toothpaste and fluoride toothpaste. So same thing is done. I'll do 0.25 grams of the xylitol toothpaste and fluoride toothpaste, let it sit, rinse, and brush, and repeat for 12 weeks. So with that, in order to measure the change of enamel remineralization, I'll be using a cannery system, which I don't know if any of you have seen this in the dental offices yet, but this is very popularly used now. And it's basically an intraoral camera that shines a pulsating laser light onto the teeth, or in my case, the typodonts. And this laser, when it penetrates the teeth actually emit a heat and light response that the laser picks up and brings to the cannery lab, which then gives you a number from 0 to 100 that represents whether it's a healthy enamel or if there's cracks and caries present. So the number from 0 to 100, if it's lower, it represents healthier enamel, and the higher it is, it represents cracks and enamels present in the tooth. And as you can see here, this shows the photothermal and luminescence response expected for the control group and the experimental group. So when it goes over the tooth and there's a carie present, it gives off a red and orange hue, and this represents the carie present. So like there's a breakdown of the enamel mineralization right there. And then the yellow and green represents healthy enamel. So in the control group, as you can see, there's not much red and orange because the teeth over time are not demineralized and they just remain the same. So for my expected outcomes for my specific AIM-2, I expect the purely extracted xylitol to have the biggest effect on remineralization and to show the lowest cannery numbers, which represents more remineralization. Whereas the xylitol and fluoride toothpaste, because they do have remineralization properties and they are used for remineralization, they will show a decrease in cannery number, but not as significant as the purely extracted xylitol. And then the control group will obviously show no change because no treatment is added to them. So these results are expected because xylitol has the ability to bind with the calcium, which facilitates calcium movement. And by applying the xylitol directly, it will enhance the remineralization results, resulting in a decrease in cannery number. And then same for the xylitol toothpaste and fluoride toothpaste. They will have a decrease in cannery number, but not as significant as a direct application of purely extracted xylitol. And because of this, the null hypothesis would not be supported because there is a change in the cannery number. So in conclusion, xylitol shows to be an effective alternative for fluoride without accompanying health effects such as decrease in IQ and fluorosis. And then more importantly, this experiment does have cheap resources and using parish fruits and vegetables, and because strawberries are present in Florida and easily obtainable, this makes it highly economical and feasible in Florida. So for my future plans, I plan to test it on real teeth in dental school rather than typodonts. And then also I would like to test different products with xylitol in it, such as birchwood and corn fiber. And I would like to test pure xylitol versus xylitol in chewing gum. And then I would like to thank Southeastern University, Dr. Franklin, my advisor, Dr. Melba Horton, for meeting with me every week for a whole year, and my friends and family and Jesus. And these are my references. Any questions? Bella.
So you mentioned that dental fluorosis
Mhm.
was a negative of using fluoride. Can you talk more about like, what that is, the implications of it?
Yeah, so dental fluorosis is actually a cosmetic condition like with your teeth. So with the overexposure of fluoride, it causes a hypomineralization on your teeth, which creates like an opaque white appearance on your teeth, which I actually have myself, and is the reason why I did this entire project, because I started researching fluorosis and the causes of it and found out that it's because of fluoride. And then further, I found out that there's more issues with fluoride. So it's something that can't be reversible because it is cosmetic unless it's very surface. But if it's done deeply, it's just basically like a scar on your teeth. So that's dental fluorosis.
On that note, I was interested in why did you not compare the application of the xylitol to fluoride application? Why did you choose it in a toothpaste form rather than
Like the direct treatment?
Direct treatment of it, since you were doing the direct treatment of yours.
Yeah, that would have been smart. I think it's more easily accessible to get toothpaste than it is for me to buy a fluoride varnish because I don't have access to that. But I can just go to the store and buy a toothpaste that has fluoride and compare it to the xylitol toothpaste.
And was there, I mean, obviously, one of your aims was to be able to get it, the rotting fruit of
Mhm.
fruit. But you said it was expensive, and yet it is readily available in most sugar-free candies.
Yes.
Like that, and xylitol itself has some side effects. I didn't hear you mention it. Have you researched or do you have any of the side effects or possible cautions that you would have to use in using it?
I did research it. I don't have it written in my PowerPoint, but the xylitol extraction process that I said was expensive and hard is not the one that I did. It's actually using lignocellulosic like biomass.
But is that what they use for the gum? Like, I
Yeah,
would think
it's
it would be a cheap process that they use to put in sugar gum.
yeah, but it's done in mass, so it's like a lot harder, and it's used not something you can do like on your own and use like in a smaller setting. It's very big and does like uses different chemicals like charcoal and stuff in order to get the xylitol from it.
And what about the complications of that?
Yeah, I researched that. It's just not in my PowerPoint.
Yeah, I was just gonna say, because there are a lot of gastrointestinal problems,
Yeah.
diarrhea. I think even patients who are diabetics, because of the sugar alcohol, it has issues
Yeah.
with that as well.
I should have put that next to
Yeah,
it.
because anytime, honestly, if you ever just pick up a pack of gum, it says.
Yeah, it
it's on
does have the
the
warning
front
labels
that
on
says
it.
So I was interested, so I was looking it up, and I couldn't find any cases that said like that they were just xylitol. They all said xylitol and fluoride.
Yeah, the xylowhite one is the only one I found that has just xylitol in it. Yeah, I struggled for a long time to find that too, but because xylitol and fluoride used in combination are very effective for remineralization.
Would you maybe have another group that is?
That has both of them. That would be a good idea, but for time wise and just with my capstone, I just had to do those 2 or 5 groups. Yes, Dr. Sam.
In general, why do they add fluoride to various products in water? Well, what's the purpose of using fluoride?
Because it helps with preventing cavities, they put it in water to help do that. But because we also use it in toothpaste and mouthwashes and everything that we have it in, it's causing an excess of fluoride amount in children specifically, because fluorosis and the IQ scores, that's happening in children. Because fluorosis can't form in adults, it forms as your enamel is forming. So they add it to help prevent cavities in areas where they can't have fluoride toothpaste. But in, say, our area where we have access to all these fluoride things, we're getting excess amount of fluoride through our water drinkage, our mouthwash, and toothpaste, and then even treatments at the dentist. Yeah, it's banned in a lot of European countries.
What are some of these noticeable side effects if they don't have fluoride? You know,
I don't have that.
there's gray teeth.
Oh yeah, it would have very bad teeth. Yeah. Yeah.
So that's, I was gonna say, it is fluoridated here in Florida.
It is, yes. The amount is 0.7 milligrams per liter that's allowed in our drinking water right now. Yes,
Yeah, not
tap
like
water.
public, like, trying to
Yeah.
steal water.
I think there is a restriction on the amount that the bottles can have as well in Florida. I don't have the exact number.
If you're going into dental school,
Mhm.
you already know about the cons, fluorine, fluoride, and whatnot. What do you think at your end and, you know, other dental people, future dental people can do as far as regulation goes?
They could go to like the politicians, or for me, like I found out that it's actually kind of easy to contact people in your area that have to do with, say, the water systems or plumbing, just anything like that. You can contact someone in your area to do that. So in the future, it would be easy to try and talk to the politicians or maybe someone who does run the water and be like, hey, I've seen these negatives with fluoride. Do you think we could figure out something to decrease the amount in water or ban it completely since we have access to a bunch of fluoride products? Sarah? So saliva actually has calcium and phosphorus in it. So with the xylitol, when it's increasing the saliva, it's increasing the calcium and phosphorus flow onto the teeth. So by doing that, it's helping in remineralization because calcium and phosphorus, as I said in the, it's too far away, the very first image that showed the calcium and phosphorus going into the tooth, that allows the remineralization. So that's why it's important. Any other questions? No? Okay, thank you. I need water in the worst way. So good.